Okay. What I was telling, see the materials you have to supply, some means proteins or some other things like oxygen it's needed. Okay, some other, so many things are needed for the normal activity of a cell. Similarly, all the waste materials produced by the cell during the metabolic activities has to be removed. Okay, so all these things, well, the only way is to pass through the membrane. Okay, so either directly through the membrane or through some proteins. This is what we have seen. Okay, but how actually that it takes place means it is through two ways I told. Okay, one it is active transport and another one is what, passive. Okay, passive means that one takes place without any expenditure of energy, whereas active transport needs energy I told. So in that, diffusion, osmosis and facilitated diffusion, these are the three types of passive transport happening across the cell membrane, and that is what I have explained you already. Okay, so
Ma'am, have
now
you shared screen?
no, not it, not it,
Okay,
because I'm going to start with a video. That video, it will be like a means a summary of all the active transport. So that is why I'm not sharing the screen. Okay, I
okay.
haven't shared. I will start directly with the video. Okay. So let me just see. See some what all I have given in the PPT and what all I have taught you. Apart from that, some extra points will be there in that video. Okay, that it will be useful to you people. Okay. So just means I have a just listen carefully. Okay, I will just play you a video now which covers the active transport part. Then I will pass, I will explain you what is passive transport. I will show you a video now. Then I will explain you what is active transport, and that also I will just explain it to you with the help of a video. Okay, so now I'm going to play a video.
Ma'am, could you play the video, ma'am?
Not it.
My screen is not shared.
One second, one second, ma. Yeah, is the screen visible?
Yes, ma'am.
Yes.
Is it audible?
So how does
Yes, ma'am.
Okay, okay then.
substances move into and out of a cell? Well, cells have a plasma membrane that acts like a fence, and this plasma membrane is what regulates what moves into and out of a cell. The membrane is comprised mainly of phospholipid molecules and has protein molecules scattered throughout its structure. The phospholipid molecules have a polar hydrophilic water-loving head and two nonpolar hydrophobic water-fearing tails. The membrane is formed as the heads face out and the tails face in, forming a bilayer, as you can see here. The inner part of the cell, called the cytosol, is here, and the outside of the cell is surrounded by a fluid called interstitial fluid. Substances move into and out of a cell through several different processes called membrane transport. There are two main processes: passive transport processes and active transport processes. The main difference between the two is that passive processes do not require energy expenditure, and active processes do require the cells to expend energy. Let's start by looking at the passive processes, which include simple diffusion, facilitated diffusion, and osmosis. Diffusion is the movement of a substance from where it has a high concentration to where it has a low concentration, or the tendency of a substance to spread out evenly over a given space. For instance, when a sugar cube is dissolved in water. Over time, the cube will dissolve and eventually spread out evenly in the water until it reaches equilibrium. Diffusion occurs down a concentration gradient, which is a difference in concentration of a substance between two areas. So the sugar molecules will move from an area of high concentration to an area of low concentration. Cellular diffusion is when diffusion of a solute, which is a dissolved substance, occurs across the plasma membrane from an area of high concentration to an area of low concentration. This is dependent on the concentration of a substance in the interstitial fluid outside of the cell and the cytosol inside of the cell. This can occur through simple diffusion or facilitated diffusion. Simple diffusion occurs with solutes that are small and nonpolar. By being nonpolar, they can move in between phospholipid molecules that form the plasma membrane because the interior region of the membrane is nonpolar. Some of the materials that move by simple diffusion include the gases oxygen and carbon dioxide and small fatty acids. So if there's a higher concentration of oxygen O2 molecules outside of a cell, they can move down the concentration gradient across the membrane without assistance and into the cell as long as the concentration gradient exists. And if there's a higher concentration of carbon dioxide CO2 molecules inside a cell, they can move across the membrane without assistance out of the cell into the interstitial fluid. So again, simple diffusion is when nonpolar molecules pass directly through a membrane. The second type of diffusion is facilitated diffusion. This applies to solutes that are small and either charged or polar. Because these solutes are polar, the nonpolar phospholipid bilayer blocks them from passing through the membrane and into or out of the cell by simple diffusion. However, they can pass into and out of the cell with the assistance of plasma membrane proteins through a process called facilitated diffusion. There are two types of facilitated diffusion: channel-mediated diffusion and carrier-mediated diffusion. The difference between the two is the type of transport protein used to move the substance across the membrane. Channel-mediated diffusion is when an ion, which is a charged particle where its total number of electrons does not equal its total number of protons, giving it a positive or negative charge, moves across the membrane through a water-filled protein channel. Each protein channel is typically specific for one type of ion, and there are two types of channels: a leak channel, which is continuously open, and a gated channel, which only opens due to a stimulus and only stays open for a fraction of a second.
See, listen, actually, this gated channel, leak channel, all those things, no? Later in active, when we will be studying about action potential, we need all these things. Okay, see, all these channels are just nothing but proteins only. Okay, but the way how it is going to transmit the particle inside, that varies. Okay, so based upon that only, they are giving all the different sorts of things they are giving. See, either channel, that is what channel is, that is what now they are explaining. Leak channels means it's a kind of protein which will allow the flow always, it will be open always. Okay, but the same type, if it is going to have a small gate, and the gate will open only when it receives a message. Okay, when it receives a stimulus. Okay, so those kind of channel will be called as what? Gated channel. Okay, don't get confused. This is all just nothing but proteins present in the phospholipid bilayer. I have told you, no, the proteins, it will act as transporters. Okay, so one kind of a protein, okay, it will act like a channel to allow the substances to pass through.
So for a sodium positive ion, it can pass through a sodium positive leak channel continuously, and a gated sodium positive channel will only open due to a stimulus to allow the ion to pass through into the cell. Carrier-mediated diffusion involves the movement of polar molecules, such as simple sugars or simple carbohydrates, and amino acids.
See, and one more type of a protein is what is the carriers. They will be called as carriers. See, channel carriers, everything it is protein only, but the that's what I told. No, the way how it is going to transmit the particle in channels, easily it will pass through. Okay, but carriers, they will change those proteins. They will change their shape accordingly to transmit these particles. So carriers are one kind of proteins and channels are one kind of proteins, but both helps in what? Facilitated diffusion only.
Across the membrane. This is accomplished by a carrier protein, which actually changes shape in the process. For instance, glucose binds to a carrier protein, which changes shape and moves the glucose molecule to the other side of the membrane. Now for osmosis. Osmosis is the passive movement of water through a selectively permeable membrane. This occurs when there's a difference in concentration of water on either side of the membrane. This can happen in one of two ways. Water can slip between the phospholipid molecules that make up the plasma membrane, or through integral protein water channels that are called aquaporins. The plasma membrane is not permeable to most solutes, such as charged polar and large substances. So for example, one side of the membrane, the cytosol or interstitial fluid, can have more solutes than the other side. Let's say the interstitial fluid has 3% solutes and 97% water, and the cytosol side has 1% solutes and 99% water. In this example, water will move down its concentration gradient from the 99% cytosol side to the 97% interstitial.
So see, already if you remember, I have told you water can move through two ways, okay, either directly across the membrane or through these proteins also. I told if it is through the protein, no, the rate of diffusion, okay, it's more, okay, more faster. The water molecules can get inside or outside depending upon the concentration gradient. That is what they are showing. Okay, either it can flow through the semi-permeable membrane, that is your phospholipid bilayer, or through proteins. Okay, and the name of that protein is what is called as aquaporin. Okay, aquaporins means they are the proteins present in the what is that phospholipid bilayer, which allows water molecules to pass through.
Interstitial side to achieve equilibrium. Now let's look at active processes. Active processes require the use of cellular energy for membrane transport. There are two types of active processes: active transport and vesicular transport. Active transport is the movement of a solute against its concentration gradient, or going from an area of low concentration to a place of higher concentration. Vesicular transport is the transport of large substances across the plasma membrane by a vesicule, which is a membrane-bound sac filled with materials. Active transport has two types.
See, just watch this active transport also. Later I will explain you. Okay, just go through this, like this video, because here they will show you what is endocytosis, exocytosis also. Just know and keep, okay?
Primary active transport and secondary active transport. In primary active transport, cellular protein pumps, called ion pumps, move ions across the membrane against their concentration gradient. Let's take a look at an example. Here we have a membrane along with a sodium positive and potassium positive.
Are you able to see the PPT?
No, ma'am.
Yes, ma'am.
Yes, ma'am.
Is
Yes,
visible.
ma'am. Yes, ma'am. Yes, ma'am. It's
Yeah, better. Let me first explain you, okay? And then we will just to see. Okay. So here, what is your okay? What is active transport? Okay. So here, as the name suggests, there will be a movement from what? Uh, Normally we have seen from higher to lower. That happens automatically. But lower to higher means that definitely needs what? Energy. Okay. So the movement of substance against a concentration gradient. Okay. From a region of lower concentration to a region of higher concentration using an input of energy is what is called as active transport. Okay, so in normally biological system, if you take energy itself means what is the thing, what actually provides energy? You would have heard this term, right? ATP
ATP.
molecules. ATP molecules are the one which is going to provide the energy. So what is this ATP? It is adenosine triphosphate. Okay, so this is what is going to provide energy for this particular movement to take place. Okay, so the process transports substances through what? A membrane protein. Obviously, this is also through what? A protein only. Okay, it cannot happen directly through the phospholipid bilayer. Okay, so the movement of substances is selective via the carrier proteins and can occur into or out of the cell. Okay, so from outside to inside also it's possible, or from inside to outside also it is possible. Okay. So such a type of transport is called as what? Active transport. Okay. So the very familiar example normally what they will give is this sodium potassium pump. Okay. Let me explain you what is this sodium potassium pump. Okay. See, normally just see this diagram. See, what they have given, I have already told you for a normal functioning of the cell, the ions play a very important role, particularly nerves and nerve cells and all. If you take in neurons, okay, if it has to communicate any information for communications, okay, so this sodium and potassium ion concentration plays a very important role. Okay, see, normally in a normal resting cell, okay, see potassium ions, okay, inside, are you able to see here? See which type of ion is more inside the cell? The concentration of potassium is more inside the cell, whereas outside, okay, in the extracellular space, you will be having more number of what? Sodium ion. Got it? This you have to remember. Okay. See, in intracellular space, you will be having more and more what? Potassium ions. Okay. Whereas in the extracellular space, you will be having what? More and more sodium ion. Okay. See, now in order for action potential, I will tell you what is action potential also. To maintain, it requires a movement of sodium and potassium across this, what is this, phospholipid bilayer. See what actually it is needed, no? See, from inside, okay, there will be a movement of sodium ions to outside, okay, and from outside there will be a movement of potassium ion to inside. Okay, got it? See, potassium is more inside, okay, and sodium is more outside. But sodium ion from the intracellular space where the concentration of sodium is very less, from inside, sodium it will move towards outside. So it's moving from what? A lower concentration to what? A higher concentration. Similarly, potassium, which is very less in concentration outside, it will move inside. Okay, this actually happens for a purpose. Okay, but what actually is the purpose? That you will come to know in what? The later classes only. Okay, because if I explain you that all those things means, then that is what is action potential. So you just need you just understand now. Just for action potential to happen, this movement of sodium to outside and the movement of potassium to the intracellular space is required. Okay. See, but this has to happen against what? The concentration gradient. Okay, so how actually it happens means there is a carrier protein, there is a special carrier protein called as sodium potassium pump. Okay, this is what is the sodium potassium pump. Okay, so this is a protein only. So what will happen, you know? See, this protein, it has a provision for 3 sodium ions to get attached in a single go. Okay, so 3 sodium ions from inside, it will get attached to this particular what sodium potassium pump. So once when it gets attached, and when an ATP, it comes and associate with this, it just gets dissociated into what? An ADP and a phosphate. Okay, see ATP, what it will happen, you know, ATP, triphosphate, right? In that one of the phosphate, it will get attached to the sodium potassium pump, and the remaining ADP, it will just move away. Okay, that means what? It's providing energy. Okay, this phosphate, it gets attached to the sodium pump. So as this phosphate gets attached to this sodium pump, the 3 sodium ions which got already attached to the sodium pump, it will be expelled outside. Okay, so as it gets expelled outside, it will be having the provision. See, the shape of the sodium pump itself, it will be like that. 3 sodium can attach and 2 potassium can attach. Okay, for every 3 sodium ion moving out, 2 potassium ions will come inside. So obviously if you see, this charge difference will come. This charge difference is what is needed, okay, for communications. So now when the 3 sodium ions, when it gets outside, 2 potassium, it comes and gets attached here. Okay, so now again, what will happen? This phosphate, whatever it was attached, it get detached, and now these 2 potassium ions, it will be just transported inside the cell. Okay, see, for this also, let me show you a video. In video, it will be very, very clear. You can easily understand. Okay, see, later that, after that, if suppose if you are having any doubts, you can ask me. Okay, so what is this active transport? Here, there is a movement of substances from a lower concentration to a higher concentration. Okay, so it happens with the help of what? Energy. That energy is provided by what? Adenosine triphosphate. Okay, so best example is sodium potassium pump. Okay, sodium potassium pump, it's a protein which is going to transport sodium from inside the cell to the outside, and at the same time it will transport 2 potassium ions from outside to inside. Okay, so this happens with the help of what? ATP. Understood? This is what is active transport. Now let me show you a video which clearly explains you how this sodium potassium pump will work. Are you able to see the screen?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Let's see, have you understood what is active transport now? Do you have any doubts in this?
Understood.
Any doubts?
Ma'am,
Means
potassium have low concentration.
Potassium have
Low concentration.
low concentration.
Means there
outside.
are chemicals. Yes. Yes.
See, outside the cell, normally the potassium concentration will be less. Okay. So what actually it is needed is, see, if you have to communicate impulses, okay, neurons communicate impulses, right? So how actually it is communicated means through the generation of these charges only. Okay, normally the impulses, when it reaches any cell, it creates a charge. Okay, so for that particular purpose, what actually will happen, you know, potassium ion from outside it will try to get inside, okay, and sodium ion from inside it will try to move outside
Yes,
against
ma'am.
the concentration gradient.
This process is active process. That's right.
Yes, because this happens with the help of ATP, right?
Yes, ma'am.
So it is a active process. Got it?
Yeah, ma'am.
Yeah. So see, suppose if they're asking you to differentiate what is facilitated diffusion and active transport, how will you justify? Anyone? Facilitated diffusion and active transport.
Ma'am, isn't it just the energy?
Isn't it?
For active transport, you need energy.
Yeah.
And for the facilitated diffusion, you don't need energy.
Yeah,
It
we
just
don't
happens
need.
due to the difference in.
Yeah, see, only one common thing is there actually facilitated diffusion also, it happens through what? Proteins only. Okay, let it be a channel protein, let it be a carrier protein. That also it takes place through what? Protein. Okay, but that doesn't require any energy, and the that is the direction. Okay, it will be from what? Higher concentration to a lower concentration. Understood? So the only common thing between the facilitated diffusion and this sodium potassium pump is what? It is happening through what? Protein present in the phospholipid bilayer. Okay.
Ma'am, I have a
Yeah.
active process is only through proteins.
Yeah.
Okay. Yes.
Protein zone. Okay. So with this much if you know regarding the transport, it is enough. Okay. So I will just summarize. I have given it in the form of a diagram. So just let me explain. Are you able to see the slide?
Yes, ma'am.
Yeah. So this is the concluding file means what is that slide for your active and passive transport. Okay. So diffusion, it will move from a region of what? Higher concentration to a lower concentration. See, remember, for diffusion and facilitated diffusion, this region is what? Higher solute concentration and lower solute concentration. Okay. But osmosis, you see, they have mentioned this region as what? Lower solute concentration. Lower solute concentration means obviously it is what? Higher. Water is more. Okay. So this side water is more and here water is less. So obviously there will be a movement of water across the membrane. Okay. So what is active transport? It will take place through a protein, and it happens from a region of what? Lower concentration to a region of what? Higher concentration with the help of what? ATP. Okay, so this summarizes all the 4 different types of transport. See, apart from that, there are so many things. Just they have mentioned, no, you have even in active transport also there are two types: primary transport, secondary transport. Primary transport means whatever we have seen. This is what is a primary transport. Secondary means from primary it will take energy and it will try to make another kind of a transport to happen. Okay, all those things it's not needed for you people. This much if you know, it is sufficient. Okay. So now what we have seen we have started from the cell, right? First we have seen the structure of the membrane, plasma membrane. Okay, so how actually it is, how is the phospholipid, what all it will allow, what all it will not allow, and we have seen the proteins, what are all the different function performed by a protein. Now we have seen how actually the transport happens across the membrane. Okay, see now let me slowly start with 111 organelle. Okay, so in that first comes this, first one is the nucleus. Okay, so what all you know about the nucleus? What all you remember?
Ma'am, one small doubt.
Yeah.
For the exam point of view, ma'am, like biology
Yeah.
paper,
Yes.
if we just do your PPT thoroughly, would it suffice, or should we refer other books as well?
This much is enough, but to get a complete idea, you have to go through books. Books are any material related to this, because see, so many things I'm just giving you. You will not be able to take down all those things also, right? Here, all important points only I have given. So if you refer it, if, along with books means it's good, but from syllabus point of view, strictly if you follow till whatever I have given in the PPT, it's enough.
Okay, ma'am. Thank you.
Yes, so nucleus means, if I say nucleus, what all you remember?
It's the brain of the new cell.
It's the brain of the cell, yeah. So nucleus is what is called as the brain of a cell. Then any other points?
It also consists of the genetic material.
Yeah, it consists of the genetic material. Yeah, then yeah, nucleus. So you forgot. So see, it's a very important thing, right? Nucleus plays a very important role among all the cell organelles. This is the one which is going to control everything, right? So what all you remember? Others?
And it's mainly made up of proteins.
Proteins. Okay, then.
Ma'am, last I studied in 10th, ma'am.
So nothing you
Yes.
remember.
No, very, very little, ma'am. Just these
Very,
many.
very little. See, one more thing here, let me just, apart from when you have studied, now you are studying biology, okay? See, now we have to understand, know how we work, how actually all of our organs work. I think you will be having that curiosity, right? You people have that curiosity or not?
Yes, ma'am.
How you're able to
Yes,
talk, how
ma'am.
you're able to communicate, what is happening inside you, how a heart beats, how a kidney works, okay, how a liver is able to do all its job, okay, what is see, that curiosity, it will be there or not?
Yes, ma'am.
See, anything studied out of curiosity you will not forget, right?
True, ma'am.
But see, what I'm trying to tell is, at least now you just see all this and not just leave apart as a subject you have to study, you have to score mark, leave all those things. Okay, apart from that, you just see this as an this as a thing which is going to explain about you. Okay, how, just to try to relate each and everything, like how it works, how I am able to do this, how it is, how it actually happens. Okay, but because now you have come to that particular stage, okay, start thinking in that way, start analyzing in that way. Okay, so then you can make it slightly interesting at least. Okay, yeah. So and one more thing, anyhow, all these things, that is what, if nucleus means, I can teach this nucleus for 2 to 3 hours also. Okay, that much things are there under nucleus. Okay, see, but our paper, no, I already have told you, we are just going to see how biology has combined with technology, how you can use technology. Okay, so for that particular purpose, if you have to understand how technology has to be used, you should know a little bit of biology. So for that purpose, I'm just teaching you all these things. Okay, so I'm not going in depth of anything. Just you should know. See later, whichever term I'm just going to tell, you should be able to understand. Okay, oh, this is what is this like. Okay, for that purpose only, the syllabus is also framed, and I have just giving you the PPT also as per the thing. Okay, so nucleus. So this is the largest organelle in the cell. Okay, and it contains the cell's genetic information in the form of DNA. What is this DNA? See, who is this? Somebody you can you?
Deoxyribose nucleic acid.
Yeah. So what?
Deoxyribonucleic acid.
Yeah, deoxyribonucleic acid. Okay, so DNA. See, do you know what is the difference between a gene and a DNA? What is a gene? They will normally, the word it will be used, you know, gene, what is it? Gene, gene and DNA. What is gene? And what,
DNA is a little more smaller aspect, ma'am, as compared to the gene. And gene is said to be
hmm.
like, I don't know how to put it in words, ma'am. Just 1
Anyone
minute.
else? Anyone else? Okay, RS, what is this DNA composed of? Any idea?
Nucleic acid.
Nucleic acid, okay.
It's composed of 4 amino acids, no, ma'am.
Ma'am, proteins.
I forgot the names, ma'am.
No, names will leave it. 4 amino acids. Okay.
Thymine,
Those stick kind of a thing, whatever it is there in between this double helical strand, no, that are those 4 things. Okay. What about that helical strand? What is that made up of? Do you remember? Are you able to
Proteins.
imagine the
Nucleic acid.
nucleic
Genes are
acid?
made of DNA.
What?
AGTC.
AGTC is what is that is what he told. Adenine, guanine, thymine, cytosine, all those things. No, I'm talking about that helical structure.
Nucleotides.
Nucleotide. See, are you able to see here? These are what you are telling. Okay, AGTC and all. What about this double helical structure? This one, the blue one made up of?
aren't they made up of proteins?
Proteins.
Yes, ma'am.
Do you think it's made of
Chromatin fiber.
chromatin fiber? See here, so many terminologies will be there, no? DNA, gene, protein, chromatin fiber, chromosome. Then apart from that, you have one more term called as chromatid also. Okay, do you know the difference between, hopefully, I don't know whether you know or not, but later, let me tell you all those things. Okay, yeah, so now DNA, so deoxyribonucleic acid, I will tell you about all those things in later, in future classes, okay? So the presence of a nucleus is a primary factor that distinguishes eukaryotes from prokaryotes. We have seen, right, the difference between a eukaryote and a prokaryote. A well-defined nucleus, if it is having means, then that is called as what? Eukaryote. Okay, so now coming to the structure of the nucleus. Okay, so the nucleus actually it is surrounded by a membrane. Okay, so just like how cell have the cell membrane, okay, we have been studying, right, plasma membrane. The same way the nucleus also has what? The membrane. Okay, and it is made up of what? Phospholipid studded with proteins. Okay, so the same way, just like how a cell is going to have a membrane, the nucleus is also going to have a membrane, and here there will be 2 membranes, one outer membrane and one inner membrane. Okay, the inner membrane is the one which is going to cover, okay, or protect the genetic material. Okay, whereas the outer membrane, it will be in contact with this endoplasmic reticulum and all. Okay, so now next important one is nuclear pore. Okay, nuclear pores. Okay, tiny openings on the surface of the nucleus is what is called as the nuclear pore. So what is the main function of this nuclear pore? Means it will allow the exchange of substances to happen across the nucleus. Okay, see, whatever, if anything, see, initially what they thought that only from inside the nucleus this RNA and ribosome can move out. Okay, nothing can move from cytoplasm to the nucleus. But recently they have proved that theory to be wrong, and they have shown that from nucleus to cytoplasm and from cytoplasm to nucleus, movement is possible, and the proteins, RNA, all those things can move inside and outside what? The nucleus, and it happens through what? This nuclear pores. Okay. Next is called, next is what is chromatin. So this is what is your DNA. See, normally, see, chromosome, okay, chromosome, that word becomes important only during the cell division phase. Okay, when the cell is going to divide, right, at that time only this formation of chromosome takes place. Otherwise your DNA, okay, it will be in the form of what? Chromatin. Okay, what is this chromatin? It's just thin long strands of DNA along with some proteins called as histones. Okay, see this, that protein is having a special name called as what? Histone. Okay, DNA along with some proteins, okay, in the form of thin long strand is what is called as a chromatin fiber. In a normal state, okay, the DNA, it will be in the form of chromatin only. Only during cell division they all will condense, okay, together to form what? Chromosomes. Okay, so next, one more important thing is what is nucleolus. Okay, so this nucleolus, it's what actually it is the main function means, it makes a specific RNA. See, what is this RNA?
Ribonucleic acid.
Yeah, see, DNA means what? It is deoxy nucleic acid, deoxyribonucleic acid. RNA is what? Ribonucleic acid.
Ribonucleic acid.
Okay, so the nucleolus is what is the main function of the nucleolus is the synthesis of this RNA. Okay, see this RNA, no, there are so many different types of RNA. rRNA, okay, ribosomal RNA is there. mRNA is there. Okay, mRNA means messenger RNA. Similarly, you have something called as tRNA also. Okay, later you will study about all these types of RNAs. Okay, so this rRNA called as ribosomal RNA is very, very important in the synthesis of proteins. Okay, so this is the one which is going to carry the sequence of amino acid needed for a protein molecule to get built. Okay, so this is made in nucleolus, and it comes outside the nucleolus and it reaches the cytoplasm, and with the help of ribosomes proteins will be synthesized. Okay, so nucleolus is this it's a portion in the nucleus where actually the synthesis of particularly rRNA will happen. Okay, so the nucleus, okay, nucleus means nucleus is inside, okay, the cell. It's completely isolated from the cytoplasm by what? Membranes. Okay, nuclear membrane. You have two membrane, inner membrane, outer membrane, and these membrane protects the nuclear materials. Okay, that is, it keeps it isolated from your what? The other cell cytoplasmic organelles or cytoplasm. Okay, see, even this nucleus is filled with a gel kind of a thing, or watery kind of a thing called as nucleoplasm. Okay, just like cytoplasm, you have something called as what? Nucleoplasm. In this nucleoplasm is it means it's suspended only all this chromatin, nucleolus and all. Okay, so you have a jelly like thing called as nucleoplasm. You have a nuclear envelope, you have nuclear pores on the surface of the nucleus, which facilitates the movement of materials, okay, between the nucleus and cytoplasm. See, your genetic material, now it will be in the form of what? Chromatin fibers, and you have something called as nucleolus, okay, which actually plays a very important role in the synthesis of RNA. Okay, and do you know, see, whether all cells contain nucleus? Do you know any exception for this? See, you prokaryotes, you leave. Prokaryotes, they don't have well-defined nucleus. You have some genetic material in the form of DNA which freely floats. Okay, that is different. See, even in eukaryotes, even in our human body, if you take red blood cells, you know, no, RBC. You know, right? What are RBCs? Red blood cells. Okay, a matured red blood cell, it will not have any nucleus. Okay, so similarly, you have some skeletal cells where there will be multiple nucleus. Okay, so you have more than one nucleus also in certain types of cells, and you don't have nucleus in what red blood cells also. Okay, so majority there will be what, only one nucleus. So now you can see here, see this is the nuclear envelope. The dark blue one is the nuclear envelope. This strands, okay, of fibers are just nothing but your genetic material in the form of chromatin, and the center one you are able to see, no, that is what actually it is, the nucleolus. Okay, see, and I told you, no, there are two membranes. Are you able to see one membrane? It covers this, and the outer membrane actually it is associated with what? The endoplasmic reticulum. Okay. See, and pores, are you able to see this one? See the yellow ones on the surface, these are actually the pores present on the surface, which allows the exchange of materials. Okay, so with this, let me stop. In next class, let me explain you what actually is this chromosome. Okay, what are the functions of nucleus? All those things we will see in what? The next class. Okay, I think I have uploaded all these things already in the last class. Okay, so for Thursday's class, you just go through and come once, full completely. Okay, then only you will be able to understand the remaining things. Yeah.
Ma'am, this PPT is not there in the links. This PPT is not there in the links, ma'am.
It's not there.
Mine
Cell transport, I have uploaded one. I think it is there. You just check once, okay? Cell transport.
Full PPT was not there. There is only cell structure. There was only cell structure and cell structure plasma membrane.
Ma'am, cell structure was the last you uploaded, ma'am.
Okay, okay, now I will do. Okay, yeah, cell structure introduction and plasma membrane only I've given. I will do it now. Immediately, yes. I will just upload it. Okay.
Okay, ma'am. Thank you, ma'am.
Yeah, thank you.
Thank you, ma'am.
Yes, thank you.
Thank you, ma'am.
Thank you, ma'am.